Hello folks, and welcome to video 2 on respiration, in which I explain the process of aerobic respiration. Organisms that can use oxygen to help break down organic compounds are considered aerobic organisms. Some organisms can only do anaerobic respiration, and they're called obligate anaerobes, while others can survive well with or without oxygen and are termed facultative anaerobes. These two groups of organisms are typically primitive and are bacteria and some fungi. Yeast, a fungus, is a facultative anaerobe. In this video, I'll focus on aerobic respiration. Remember that it involves 3 steps: glycolysis, the citric acid cycle, and the electron transport chain. I think it will do you some good to quickly review the process of glycolysis. If you haven't had a chance to watch my video, part 1 of cellular respiration, glycolysis, you'll definitely want to go back and watch that. Respiration, aerobic or anaerobic, starts with glycolysis. You'll remember that it starts with 1 glucose molecule and that the end products are 2 pyruvate molecules. The net gain is 2 ATP. This process happens in the cytosol of the cell. After glycolysis, and if oxygen is present, many organisms will perform aerobic respiration. The pyruvate will be transported into the mitochondria, the powerhouse of the cell, and enter the next series of reactions to take more energy from the pyruvate molecules. Mitochondria are the ATP factories of eukaryotic cells. They're found in both plant and animal cells. Their fuel is pyruvate. It's important to review a bit of mitochondria anatomy here. It's a double membrane organelle with an outer and inner membrane. The inner membrane, referred to as the cristae, is highly folded to increase surface area and maximize the ATP production process. There's an intermembrane space, and the matrix is the inner space. Both spaces are filled with cytosol similar to that of the cell. Now let's get to the process. The first step, glycolysis, is taking place in the cytosol of the cell, breaking down glucose to produce some ATP and 2 pyruvate molecules. Next, to be described in greater detail, is the citric acid or Kreb cycle. This is taking place in the mitochondrial matrix. This cycle is named the Kreb cycle after a wonderful man by the name of Hans Krebs, who, as a young adult of Jewish heritage, escaped Nazi Germany in the 1930s and settled in England. While researching aerobic respiration, he discovered the citric acid cycle. The last thing to occur is the electron transport chain. It's here where the oxygen is necessary and where most of the ATP is made. So let's dive into the citric acid cycle, shall we? Here you see the process all laid out. You see it's a metabolic pathway that is actually a cycle whereby some products are reused in the process. If you're like me, you'll need to learn this process in small steps. So let's cover up most of it and take a little bit at a time. Remember, numerous enzymes are involved but are left out of the explanation. Here you see 1 pyruvic acid molecule, a 3-carbon compound. First, an enzyme facilitates the oxidation of pyruvic acid. NAD is an electron transport molecule. It strips off electrons and a hydrogen atom. A carbon and 2 oxygens are released to form carbon dioxide. Carbon dioxide is a waste product that the cell gets rid of. You breathe it out. Next, a coenzyme, called coenzyme A, joins the newly formed 2-carbon compound to form acetyl-CoA, which combines with a 4-carbon compound to make citric acid. The coenzyme A is released at this point to be used again. Citric acid is a 6-carbon compound. The citric acid progresses through the reaction. Carbon dioxide is released again, and NAD oxidizes the reactants, leaving a 5-carbon compound. In the next step, another CO2 is released and another NAD does some oxidizing, and one ADP is phosphorylated into one ATP. This leaves a 4-carbon compound. But wait, there's more. Enzymes help to oxidize the molecule even more to strip off more high-energy electrons. FAD, flavin adenine dinucleotide, another electron transporter that's like NAD, and another NAD take those electrons and hydrogens off. The 4-carbon compound is then available to re-enter the process. One molecule of glucose causes 2 turns of the Krebs cycle. Here's a diagram in a bit more detail. Pretty busy place, that mitochondria matrix. All the enzymes are left out of this diagram too. Now for the last part, the electron transport chain. This process takes place on the cristae of the mitochondria. That's the inner membrane of the mitochondria. Here you're going to see the phospholipid bilayer of the cristae, and embedded in it are enzymes that will transport electrons from one place to another. Hence, it's called the electron transport chain. The names of the enzymes are not important at this point, but what they're doing is important. They use the energy of the electrons to power the active transport process of pumping protons, these hydrogen atoms, from the matrix into the intermembrane space. You see that the source of the electrons and the hydrogen atoms are the NADs and the FADs that were produced in glycolysis in the Krebs cycle. The electrons are picked up at the end by an oxygen and combine with hydrogen to make water. The high concentration of hydrogen ions in the intermembrane space have a lot of energy too, because of their high concentration. So at this point, are you asking, what's the point of the electron transport chain? No? Well, go ahead and say it. What's the point of the electron transport chain? Well, it's not just to make water. That high concentration of hydrogens can do some work. Here's an enzyme in the cristae that has an important role of phosphorylizing ADP into ATP. It's called ATP synthase. The electron transport chain created the proton gradient that powers this enzyme to do the endergonic process of making ATP from ADP. Those hydrogen ions, or protons, provide what is called the proton motive force as they move through that enzyme. Here you see the phospholipid bilayer of the mitochondrial cristae and the embedded enzymes. This is the electron transport chain. In the mitochondrial matrix, you have the NAD, which is now reduced with the hydrogen and the electrons. Here's an oxygen and some of the hydrogens. You'll see NAD move into the first enzyme, and it will release 2 electrons and the hydrogens. The energy from those electrons are used to pump the hydrogen into the intermembrane space, therefore increasing the concentration there. As the electrons move in, more hydrogens pumped across. You'll see here at the very end that the 2 electrons join up with an oxygen molecule and some hydrogen to form water. So the electrons are pulled through as they're taken off by the oxygen. Water is a byproduct of respiration. Now that high concentration of hydrogen ions has created some energy. Here, the ATP synthase allows them to move back with the concentration gradient, using that proton motive force of the moving hydrogen ions to phosphorylize ADP into ATP. That's chemiosmosis. Let's go back to this diagram here. In both glycolysis and Krebs cycle, some ATP were made directly because they were formed from the catabolism of organic compounds. The organic compounds were a substrate in an enzyme-mediated reaction. Because their energy was used to make the ATP, we say that ATP was formed from substrate level phosphorylation. In the electron transport chain, those ATP were formed as a result of the oxidation of those organic compounds earlier in the Kreb cycle and glycolysis. For that reason, we say that those ATP were formed from oxidative phosphorylation. Now let's do some accounting to see how much ATP is made from just one glucose molecule. 2 ATP each from glycolysis and the Kreb cycle for a total of 4 ATP. Also produced from one glucose was 2 NADHs from glycolysis, 8 from the Kreb cycle, as well as 2 FADHs. The process can make up to 3 ATP from 1 NADH and 2 ATP from 1 FADH in the electron transport chain. So 34 ATP come from the electron transport chain in chemiosmosis. So the 4 ATP from the substrate level phosphorylation and the 34 from the oxidative phosphorylation give us 38 ATP from one glucose molecule. Some more math will show us how efficient that is. The complete oxidation of glucose has a change of free energy of negative 686 kilocalories per mole. Through aerobic respiration, we get 38 ATP from one glucose molecule, each with a negative 7.3 kilocalories per mole of free energy. That's a total of 277.4 kilocalories per mole total. That makes aerobic respiration around 40% efficient. As energy systems go, that's not too bad. That's it for a very quick explanation of aerobic respiration. I hope that by watching this video and taking notes closely in some of the discussion and activities that we do in class, that you'll get to understand the process a little bit more closely. Until then, we'll see you back in class.